Good evening, everyone. So this is week 7, and we have switched to microbiology. So environmental chemistry has been finished. So we will talk about the microbiology, and today is the introductory lecture about the microbiology. So what we will discuss. So actually, this is based on the introduction to microbiology only, and we will get to know what do you mean by microorganism, then different type of microorganism that we have on the earth, the characteristics of this microorganism, the difference between genomic material, or you can say the genetic material of the of the microorganisms. So we will talk about basically the structure, function, and the types of microorganism in this lecture. So let's start with my first because this is very introductory session about the microbiology, so the questions are very simple and you people can comfortably give me the answer for this. Okay, so my first question is, which of the following is considered a microorganism? And we have four options: bacteria, fungi, virus, and algae. B is only bacteria and virus, and C is only multicellular organism, and D is only virus. So, what do you think? Like, what should be the answer for this? Which of the following is considered a microorganism? You people can write in the chat box. You can give me the answer for this. Otherwise, I will also explain why and what is the answer for this. Okay. You gave me answer B, but not only just bacteria and virus, Smita, you have to include fungi, algae, everything. Like the smaller organism that we can't see. Okay, and this may also include fungi and algae as well. So yes, so A is the right answer. Let me show you. So we have the classification of the organism that is given by Robert H. Whitaker, and in which it classified organism between two types: prokaryotic and eukaryotic. Pro means very primitive, very simple. Okay, so these are very simple unicellular organism. Unicellular means one cell organism, right? So in prokaryotic, we have one class, that is the Monera. Okay, and Monera will have mostly the bacterias. Then we have the eukaryotics. So eukaryotic may have unicellular as well as the multicellular, but you means true and karyotic means nucleus. So we have the true nucleus here. Pro means primitive, karyotic means nucleus. So we have a very primitive nucleus that will categorize into prokaryotic. Eukaryotic means true nucleus. So the organism that have the true nucleus will be considered into eukaryotic, and that has been classified into unicellular, that have the one cell organism, and multicellular, that have more than one cell. So for example, human are multicellular. And then that has been, this multicellular has been classified into whether that multicellular organism have the cell wall or not. So plants and some plants and the fungi have the cell wall, but animalia, for example, we human do not have the cell wall in our cell. So this is without cell wall. So without cell wall will be known as the animalia, and that have the cell wall, this has been categorized into two parts. The one is plenty, and another is fungi. So plenty will have all the plants that are phototrophic in nature. Phototrophic means that can cook their food itself, or you can say like in the presence of sunlight. So photo means sunlight, trophic means level, or you can say food. So with the help of light they can create their own food. So this will be known as the so these organisms will be considered into plenty. And then heterotrophic means they will not create their food themselves but have to borrow it from somewhere. So they will use the organic matter, and by which they will take the food. So this will consider into fungi. So this is the whole classification. And if I talk about a microorganism, then This prokaryotics are single celled organism, means a one cell you can't see with the naked eyes, right? So this, let me take the pen. So this will also consider into microorganism. Then unicellular, this will also consider into the microorganism, because we can't see is the with the naked eyes. So the protista, and in that protista, the plants we can see with the naked eyes. So this is not the option. This is not the microorganism, but some of the fungi we have that are very tiny in size, and we are unable to see it with the naked eyes. So this will also come under microorganism. plus plus virus, right? Plus virus. So the one, two, this two, and this three. Okay, and in this only, we also have algae. So we will talk about algae as well, plus algae, right? So these will be considered into microorganism. So microorganism is nothing but the organism that are very tiny in size, and that we can't see with the naked eyes. So these organism will be known as the microorganism. So based on that only, the answer will be bacteria, fungi, virus, and algae. I think this is clear. Let me clear this. Okay, and because I talked about prokaryotic cell and the eukaryotic cell, so you must know the difference between prokaryotic and eukaryotic. But the thing is very simple. As I told you, pro means primitive, karyotic means nucleus. So if you will see here, this is the DNA material, and this, or you can say this is known as nucleoid in prokaryotic cell, and this is scattered into the cytoplasm. This is cytoplasm. So this liquid part is cytoplasm. So in that cytoplasm we have scattered nucleoid or DNA. Okay, so this is the genetic material. So that's why it is without any wall, without any membrane, we have the DNA within the cytoplasm. So that's why this is prokaryotic. And if here we will see, this is our nucleus, the well defined nucleus. In that nucleus we have nucleolus. This is the nucleolus, and here we have all the genetic material. So this is well defined nucleus. So that's why U means true, karyotic means nucleus. So we have very well defined true nucleus. That's why this is the prokaryotic. So if you will see here the difference, they are smaller and lack of defined nucleus, but here we have the well defined nucleus. Then organelles. So here we do not have the membrane bound organelles. So organelles like this lysosome, centriose, Golgi apparatus, and this mitochondria. So these are known as the organelles. So these organelles are absent in prokaryotic cell, but we have it in eukaryotic. Okay, then if we are talking about the ribosomes, so here, so we have the ribosomes here in the eukaryotic as well as in the prokaryotic cell. Okay, so ribosome is common in eukaryotic as well as in prokaryotic, but there is a different specification of this ribosome. So here it is 70s and here it is 80s. Okay, then here in prokaryotic we do not have well developed cytoskeleton, but here we have dynamic cytoskeleton, and this is used for the mitosis and meiosis, so for the cell division, right? Then because here in prokaryotic, what happen if there is the one bacteria, that one bacteria will be break down into two and will give you the two bacteria. So this is known as the binary fission. We will talk about this a later. But in the eukaryotic cell, what we have to do is, first we have to duplicate the genetic material, and then we have to double our mitochondria, and then chromosome, and then we have to divide it. So these are known as mitosis and cytokinesis. So such kind of advanced method we have in the eukaryotic, but for the reproduction, in prokaryotic we have just the binary fission. So this is the basic difference between prokaryotic and eukaryotic. If there is any doubt, you people can write me in the chat box, so I will try to clear your question or query if you have. Okay, the next question is on virus. In last question, we got to know that virus is the microorganism, but you have to tell me some specification of the virus. So I have four options. Virus are considered living organism because they can produce on their own. The B is virus are non-cellular and require a host to replicate. C is virus are single-celled organism, and D is virus are a type of bacteria. Let me see who gave me answer. Okay, Ishmita gave me B answer. Yes, the answer is B, and let me tell you why. Uh, if I will go to if I go to my first option, bacteria are considered living organism because they can reproduce on their own. No, they can't produce their own. They must have some host. So host means, so for example, if some bacteria is infecting the human body, so human will be the host. Okay, and that bacteria cannot reproduce until they don't have the host. So they must have the host for the reproduction. Okay, so that's why this is not the right answer. Then as you told, like B is the right answer. Then virus are non-cellular. Why they are non-cellular? Because they do not have the cell structure. They just have the genetic material. Okay, they just have the packed genetic material. They are not considered cellular organism. So that's why these are non-cellular, and as we talked, these require a host to replicate. So this is the right answer. Then virus are single celled organism. They are not cellular. Then how they can be single celled organism? So this is again not right answer. And virus is very different from the bacteria, and we will tell how this bacteria and virus can also interact if you do know about the bacteriophage. So bacteriophage are the virus that can eat the so this is also not the right answer. Okay, so let me go to the explanation. Uh, okay, so I have three cellular structure, or you can say the structure of prokaryote, eukaryote, and virus. So if I will go to the prokaryotes, then prokaryotes have the genetic material in the nucleoid. Then they do have the cell wall. They do have the plasma membrane, and this is the flagella. This is the flagella. So they have the flagella. In eukaryote, the feature is that we do have a lot of organelles. For example, Golgi body, for example, mitochondria. In plants, we have the chloroplast. So we have a lot of organelles. That organelles we do not have in the prokaryotes. Okay, and if we will see here the virus, they are not cellular. This is not the cell, but what they have is they just have the genetic material, and this genetic material is covered with some envelope, and that envelope have protein. That protein will bind to the host, and that protein will help to bind to the specific host. For example, like some bacteria can't infect to the human, but they do infect to some different animal. So that protein will detect what is my specific host. So the virus is not cellular, but that just have genetic material covered by some envelope, and that also have some protein. Okay, and they have to hijack the multicellular organism to replicate itself, right? Replicate means it has the genetic material. Let's say this genetic material is DNA. If that virus has to duplicate its DNA, so we have one DNA, then if the virus needs two DNA, that it has to hijack the cell of host. Okay, after hijacking the machinery of that host cell, it can be replicate. So replicate means one DNA to another DNA. So we will have two DNA, and this is known as replication. Okay, so this process is known as the replication. So that's why the B answer was right. Let me go to the next question. I explored about this prokaryote and eukaryote. So the question is now, which group of microorganisms are known as prokaryotes? Then we have four options again: fungi and bacteria and archaea, virus and algae, and protozoa and fungi. Okay, we are talking about prokaryotes, Smita. So prokaryotes means that do not have the true nucleus. Okay, that do not have the true nucleus, that have the false or very primitive nucleus. So pro means primitive, karyotes matlab nucleus. So they have the false nucleus. And if I show you the classification here, so we have the prokaryotes, we have the eukaryotes, and in eukaryotes we have the fungi. Okay, because these have the true nucleus. Fungi have the true nucleus, so these can't be prokaryotes. Prokaryotes will come under this Monera, and Monera have archaebacteria, archaebacteria and eubacteria. So archaea means something false bacteria, and eubacteria means true bacteria. Okay, so this will have bacteria and algae as well. So the answer, let me show you. So the answer will be bacteria and archaea. So we have prokaryote means that have the primitive nucleus, and fungi have the true nucleus. So this will not be the answer. Viruses are different, like they are not considering into prokaryotes, they are just microorganism. And because these viruses do not have the cellular structure, so these are not the part of the prokaryotes. Again, fungi is again not a prokaryote but the eukaryote, it has the true nucleus. So this is again not the answer. So that's why bacteria and archaea will be the right answer. Let me show you some of the explanation here. So we have the microorganism. Then in microorganism, we have acellular that do not have the cellular organism. We have the cellular organism. And in that cellular organism, we have the prokaryotes, and it may have some eukaryote as well. In prokaryotic, we have bacteria and we have archaebacteria. Okay, and in eukaryotes we have algae, protozoa, and fungi. And the question was based on this prokaryotes only. So which of these are the prokaryotes? So bacteria and archaea are will come under prokaryotes. Okay, so just thing is that if we are classifying microorganism means that organism that we can't see with the naked eyes. So that can be bacteria, that can be virus, that can be algae, that can be fungi. Okay, but if we are talking about prokaryotes, then we are talking about the organism that do not have the true nucleus, and that will have bacteria and archaebacteria. right? And eukaryote means that have the true nucleus. Protozoa, algae, fungi, plantae, animalia, that all will have the true nucleus. So that's why this is the right one. Okay, I don't think that I should go right here in the prokaryote, but as I told you, in prokaryotes, we have this genetic material inside this cytoplasm, but do not have a very well membrane bound nucleus. So, and this genetic material is known as the nucleoid here. So, this is the structure of prokaryotic cell. Okay, as I told you, in prokaryotes, we have two major classes or major categories of prokaryotes, that are archaea and bacteria. So you must know the difference between these two. What do you mean by this archaea and bacteria? So they do have, so both are prokaryotic in nature. We talked about this. Both do not have the nucleus. The genetic material is just into the cell, not in the nucleus. So that's why they are prokaryotic. Then if we are talking about the cell wall, then bacteria have the cell wall that have one compound. This is known as the peptidoglycan. Okay, but archaea do not have the peptidoglycan. They have pseudopeptidoglycan, and also known as the murine. We will talk about this, but just keep in mind that in the cell wall, bacteria have peptidoglycan, and archaea do not have that peptidoglycan. If we are talking about the cell membrane, after cell wall, we will have the cell membrane. So that cell membrane in bacteria, these cell membrane have the ester linked compounds. Okay, and here it is ether linked, and rest of these are not very important. No, okay, the last one is important actually. Bacteria can be both helpful and harmful relationship with humans, but archaea are the kind of organism that thrives in very extreme condition, either in extreme cold condition, extreme warm condition, extreme salty condition. So these archaea thrives in very extreme environments. Okay, and non infect humans. So this is the important as well. So these are the difference between archaea and bacteria. So the most difference comes from the cell wall and membrane lipid, and like where they are living. Okay, so these are the difference between these two. Okay, which organism play a crucial role in nitrogen fixation? And we have four options: fungi, protozoa. Some of the people also call this fungi to fungi, right? But I'm calling it fungi. Fungi, protozoa, then cyanobacteria and virus. You already have given me answer C, Ipshmita. So yes, answer is C. That is the cyanobacteria. So cyanobacteria is an algae. Okay, this is not exactly the bacteria, but it is algae. And let me tell you what do you mean by this nitrogen fixation. Nitrogen fixation means we do have 75% of our air is nitrogen, that is N2. But and we organism, either it is plant or it is animal, we people need nitrogen, but we can't have this nitrogen from atmosphere directly. So what we have to do is we have to make it organic in nature. So let's say, or oxidized in nature. Let's say it is ammonium ion or nitrate, NO2, NO2 minus, NO3 minus. So this is nitrite and nitrate. So we have nitrogen in the atmosphere, but we can't use it directly. So that's why we have to convert it to some different chemical species of nitrogen. For example, ammonia ion, nitrite, nitrate. Okay, and some of the bacterias help us to do this thing. Okay, and this is known as nitrogen fixation. Okay, so we have atmospheric. Fixing the atmospheric nitrogen into some different chemical species of nitrogen is known as the nitrogen fixation. Some of the bacteria uh, do such kind of nitrogen fixation, but we also have the cyanobacteria, or this is gray blue or blue green algae. This is also known as blue green algae. This also help us to for this nitrogen fixation. And can you tell me, like, they do have very specific cell. That cell is known as the heterocyst. I show you actually. Okay, so this is the structure of your cyanobacteria. So they do have three type of cell. The one vegetative cell, the one cell that is heterocyst, and another cell is known as the um, akinete. Okay, so here vegetative cell is just for the photosynthesis, and they will produce the food. Okay, the work of vegetative cell is they have to produce the food in the presence of sunlight. Okay, but heterocyst, these are the cells that will do the nitrogen fixation. So here if you will see, here we have the carbon compound, then with the help of enzyme nitrogenase, this will, and if we have nitrogen coming from the outside, then with the help of this enzyme nitrogenase, it will convert this ammonia sorry this nitrogen into ammonium ion, or let's say the amino acid. So this is how they are fixing this atmospheric nitrogen to ammonia and to the amino acid. So this is how this heterocyst cell works. But the thing is, very important condition for this is that this nitrogenase, this nitrogenase is the enzyme that can work only in the anaerobic condition. Anaerobic condition. So anaerobic condition means that particular cell should not have the oxygen, because if you will see here, it is N2 and this is NH4. So this means we do not have the oxidation, we have the reduction. So for reduction, this cell should have the condition which do not have the oxygen, and this cell should have some specification different from vegetative cell, because for the vegetative cell, what we do, the CO2 comes under and then oxygen is produced. Okay, but here the cell wall is very different, the membrane is very different in such a way that it will resist oxygen to come under into this cell. That will create the anaerobic condition, and in that anaerobic condition only nitrogenase bacteria nitrogenase enzyme will work, and that will fix this atmospheric nitrogen to ammonium ion. Okay, so this is the, and this heterocyst cell is just the modified version of this vegetative cell only. Okay, so in cyanobacteria or in blue green algae, we have one modified cell that is known nitrogen. Okay, and for this we should have the anaerobic condition. So that's why uh, here C answer is the right one, that is a cyanobacteria. Let me clear this. Okay, we already have talked about this, but this is again one MCQ. Which component is unique to the bacterial cell wall? Cellulose, chitin, peptidoglycan, and phospholipid. So, if you people know that it is the bacterial cell wall, we have some antibiotic, right? So, if we take some antibiotic from the medicine, that antibiotic particularly particularly attack the cell wall of the bacteria and it rupture the cell wall. And if the cell wall of bacteria will be ruptured, then that's how we can kill the bacteria using antibiotics. Okay, but you have to tell me what is that specification, and that antibiotic should be very specific for that particular bacteria, right? And that bacterial cell wall for particular for that bacterial cell wall, and this bacterial cell wall is very different from the cell wall of fungi, cell wall of plants. Yes, Smita. Yes, this is C. That is peptidoglycan. So this is the compound, or this is the material that we have in the cell wall of the bacteria. Cellulose is the compound or the component of the plant cell wall. This is in the plant cell wall. If we are talking about the chitin, this chitin is the component in the cell wall of fungi. This is very specific. And if we are talking about this phospholipid is present in every cell wall, either it is for plant, either or it is for fungi, or it is for the bacteria. So this phospholipid is very common. But peptidoglycan is the one component that is unique to the bacterial cell wall. So let me show you also this diagram. So this have the bacteria. First we have the capsule, then we have cell wall. Inside it, we have plasma membrane. Inside it, we have the cytoplasm. So this is the liquid part in the cell. These were all covering capsule, cell wall, and the plasma membrane. But inside it, we have the liquid structure, that is cytoplasm. And in that cytoplasm, we have nucleoid or circular DNA. So this is the structure of prokaryotes, or you can say the bacteria. Okay, and then they also have ribosomes. This is common in prokaryotes and eukaryotes. And if we will see this one, so this cell wall structure is this one. So if we will see that cell wall, that cell wall have lipopolysaccharide. This is known as LPS. We do have porins, so the pores through which we can transport the food and all. Then we have the peptidoglycan, and we also have the lipoproteins. So, and this LPS, porine, peptidoglycan, and lipoprotein will composite the cell wall, right? And then we have the membrane. So after cell wall, we have the membrane. So this is the cytoplasmic membrane. So the structure that we have unique in the bacterial cell wall is peptidoglycan, and we can attack this peptidoglycan using antibiotics. And that antibiotic can rupture this cell wall, and that's why can stop the infection that has been caused by the bacteria using that antibiotic. So this is unique one. Let me come to the next. And this is again the same structure, but in a different way. Here also we have peptidoglycan. Just see here, peptidoglycan, and we have liposaccharides, and then we have the inner membrane. And in that inner membrane, we have the membrane protein as well. So if you are biology student, then you are well aware about the structure of cell wall and the cell membrane. Okay, the next question is, we talked about the bacterial cell wall. That bacterial cell wall have the component that is known as the peptidoglycan. Okay, but that peptidoglycan is little different in two type of bacteria. We have classified bacteria into two type: gram positive and gram negative bacteria. Okay, so you have to tell me what is the primary difference between gram positive and gram negative bacteria regarding peptidoglycan. So options are: gram positive bacteria have a thin peptidoglycan layer, gram negative lack peptidoglycan entirely, C is gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thin one. And D is gram-negative bacteria have peptidoglycan, but gram-positive bacteria do not. But we talked about that that bacteria have that cell wall, and that cell wall have the peptidoglycan. So the B answer can't be right, and the D answer can't be right, because here the but gram positive bacteria do not. This is not the right case. As we talked about, in bacteria we have the peptidoglycan cell wall. So this D is not right, and this B is also not right. The options are only among A and C. So what do you think? What is the right answer for this? Yes. Yes, answer is C only. Let me show you how. So answer is C only. Yes, gram positive. So this is the peptidoglycan layer, and in gram negative, this is the peptidoglycan layer. So in gram negative, this is very thin, and in gram positive, it is thick. Okay, but this is the cytoplasmic membrane or the plasma membrane. This is same, and we have this lipoprotein saccharide. We already talked about this. Both have this LPS, but here, if we are talking about the peptidoglycan, then gram positive have multi-layer, but gram negative have just one layer. Okay, so this is how we can differentiate between two categories of bacteria: gram positive and gram negative. Okay, which of the following statement about archaea is true regarding their cell wall? Archaea have peptidoglycan in their cell wall. Archaea lack a cell wall completely. Archaea have pseudopeptidoglycan instead of peptidoglycan. Archaea use cellulose-like plant. Okay, yes, C is the right answer. That archaea do have the cell wall, but it is not made up of peptidoglycan, but using pseudopeptidoglycan, and this pseudopeptidoglycan is also known as murine. Let me show you this. So this is archaebacteria. Archaebacteria is very similar to the bacterias. We talked about this. We just have two or three differences between this archaebacteria and bacteria. The one difference was in cell wall, that for bacteria it is peptidoglycan. For archaea this is pseudopeptidoglycan. We had another difference in plasma membrane. So one was the ester linked, and another was the ether linked. Okay, and archaea thrives in very extreme condition, and bacteria can thrive like anywhere, and they may be harmful to the human, but archaea are not that much harmful for the human. And if we will see here, these are composed of pseudopeptidoglycan, and these are nothing but the pseudomurane. Okay, and again we have the ether linked phospholipid, and then we have the plasma membrane. So this is how we can differentiate between archaebacteria and bacteria. Okay, though we do have talked about this, but if we are seeing here archaea and then bacteria difference, then archaea thrives in extreme condition. They may be thermophiles, means that thrives in very extreme temperature condition. Halophils that can thrive in a very salty condition. Bacteria has been classified into positive and negative. If we are talking about cell wall, then these are pseudopeptidoglycan and these are peptidoglycan. And these we are not talking about right now because we already have talked a little about this, but we will not touch the metabolic activity because otherwise you will get confused. Anyway, so let's come to the different. Which enzyme break down peptidoglycan leading to bacterial cell lysis? So if we need to rupture the cell or the cell wall of the bacteria, we need to have some enzyme that will break down the peptidoglycan. So you have to tell me the enzyme name. DNA polymerase, lysozyme, and reverse transcriptase, amylase. Not very much. See, okay. So let me tell you, this DNA polymerase and this reverse transcriptase are used in the genetic material. So if you want to replicate your DNA, so from one DNA you need to have two DNA. Then one of the enzyme that you need is the DNA polymerase. That means it will increase the polymerase. And then reverse transcriptase means if you have RNA, if you need to convert that RNA and you need to have DNA from RNA, then this will use this reverse transcriptase. Then we have option B and D, but this amylase is for breaking down the sugar. Amylose, have you heard about this? So this is to break down some sugar, but we are talking about the peptidoglycan. So peptido means something, protein, and then glycan, glucose. So for this we have some enzyme in the lysosome, and these are known as the lysozyme. So these lysozyme bacteria can break down, sorry, lysozyme enzyme can break down this peptidoglycan. Okay, so answer should be B only. Yes. And if I show you in animal cell, we have this. This is the lysosome. This is the lysosome. So in bacteria, in animal cell, we do have a lot of organelles. As I told you, we can have the plasma reticulum, we have the Golgi body, we have the mitochondria, and we also have some lysosome. In that lysosome we have bacteria that are known as the hydrolytic bacteria, and we have another bacteria, sorry, we are talking about enzymes, sorry. So we do have hydrolytic enzymes in lysosome, and these are lysozymes that can rupture the cell wall by breaking down the peptidoglycans. Okay, and the important thing is that if we are talking about this lysosome, these lysosome are also known as the suicidal bag, because these have a very strong enzymes that can disrupt the whole cell. Okay, and that's why it can, like, because of the lysosome that we have inside the cell itself can be died because of the enzyme that we have in the lysosome. So that's why this is also known as the suicidal bag, or the suicidal component of the whole cell. Okay, now coming to the next one. We talked about it a little bit about this. Why are antibiotic like penicillin effective against bacteria but not archaea? Archaea have a stronger cell wall. B is archaea lack peptidoglycan, the target of penicillin. C is archaea actively destroy antibiotic, and D is archaea lack a cell membrane. So what do you think? What is the right answer? So if we do have some antibiotic, and if we need to attack that antibiotic on the bacteria, and we also have some archaea as well, but this antibiotic will just work on the bacteria but not on the archaea. Why? Let me see the answer. Okay, Ipshmita gave me B answer. This is right. Yes, this is right, because archaea do not have the cell membrane made up with the peptidoglycan, but it do have the pseudopeptidoglycan. And the penicillin, the antibiotic, have the target, and that target is the peptidoglycan. Okay, so that's why the antibiotic can't antibiotic that target the peptidoglycan only cannot target archaea, but will target only the bacterias, because that the target is just the peptidoglycan. So that's why B is the right answer. So let me okay, so this is how, if we do have the penicillin and we have the bacteria, then what this penicillin will do, so we have this cell wall made up of the peptidoglycan. So this cell wall will be break down, this will be ruptured, this will be ruptured. So this is how antibiotic penicillin works on the bacteria. It ruptures the cell wall of the bacteria, and that's why this penicillin antibiotic is very effective when we need to resist or avoid or control the bacterial infection, right? So let's come to the next question. How do enzymes speed up biochemical reactions? By increasing the activation energy required for the reaction, by lowering the activation energy required for the reaction, by changing the equilibrium of the reaction, and by being consumed in the reaction. So, what do you think? What is the right answer for this? How enzyme can speed up the biochemical reaction? You people can give me the answer in the chat box out of this A, B, C, D. Okay, so you gave me A answer, but what I think is the answer will be B, because it lowers the activation energy required for the reaction. Let me show you the one graph. Okay, so we have the reactant. So let's say we have hydrogen peroxide. Okay, and we need to change that hydrogen peroxide into water and oxygen. Okay, so we need some energy, and that what energy or how much amount of energy we need is like something known as the activation energy. Okay, so if we do not have the enzyme, then this much energy we need to make a complex, and that complex will be break down and will change into water and oxygen. So this much energy is needed. But if we do have the enzymes, then only this much energy is needed. So this is lesser energy we need when we do have the enzyme. But if we do not have the enzyme, then higher activation energy is needed for the reaction to be completed. So that's why whenever we do have the enzyme, that enzymes can speed up the a chemical reaction, because the energy required is very less as comparison to the reaction when it is in the absence of some enzyme. So that's why this B answer is the right one, by lowering the activation energy required for the reaction. Okay, so let me clear this. Okay, what happened to an enzyme after it catalyzes a reaction? It is permanently altered and cannot be reused. It is consumed as a reactant. It remains unchanged and can be reused, and it becomes part of the final product. So, what do you think? What will happen to the enzyme after the completion of the reaction? You people can write in the chat box if you think that out of this A, B, C, and D, which answer you thinks suitable. You can write in the chat box. Let me tell you, enzyme work is just to uh, is just to increase the speed of the reaction, but it will not involve in the final product. It will be as it is. Let me show you. The answer is C, that it will remain unchanged and cannot be reused. So this is the figure. If you have enzyme and if you have substrate, and substrate or reactant, let's say A and B, and that A and B is combined, but then you will have enzyme. That enzyme, your substrate will form a complex with the enzyme. This is known as the enzyme substrate complex. And after that, lowering the activation energy, the substrate will be converted into product, and that product is, let's say, A and B, and your enzyme will be freed. Your enzyme will remain same, and this we can reuse again. Okay, so that's why the answer, it remain unchanged and can be reused, will be the right answer. It is not becoming the final product. So, and it is not consumed as a reactant, and it is not altered. So, so these A, B, and D are not the right answer, but C is the right answer. I think this is clear. Okay, the again is also about the enzymes, because the enzymes are very important. If you know that we have the genetic material, and if we need to duplicate our genetic material, if we have one DNA but we need another DNA or genetic material, we should have enzyme. Without enzyme we can't do anything. Enzymes is very important for the replication, for transcription, for translation. We will talk about this terminology as well, but that's why we have some question on enzyme. So which statement about enzyme specificity is true? Only enzyme can catalyst any biochemical reaction. Enzyme bind to specific substrate based on their active site shape. Enzyme bind randomly to any molecule in the cell. Enzyme function equally well at any temperature and pH. What do you think? What is the right answer? What is the true statement about the enzyme? No. Actually, the answer will be B only. Uh, okay, let me tell you why. So the option A, one enzyme can catalyst any biochemical reaction. No, each enzyme is very specific for the reaction. So for example, this is if we do have the hydrolysis reaction, means breaking down the water, or breaking down some compound into water and something, or we do have another type of reaction. Different type of reaction needs a different type of enzyme. So each of the biochemical reaction can't be happen only with the one enzyme. So A is not the right answer. Okay. Then enzyme bind randomly to any molecule. No, each enzyme is very specific to that particular reactant. That's why enzyme are very specific for that particular reaction. So this is again not the right answer. Then if you will see, in our body, if temperature is not maintained, if pH is not maintained, then enzyme will not work very efficiently. Okay, so that's why enzyme are also dependent on temperature and as well as on the pH. This is also not the answer. The answer is this one, and let me explain it with the figure. So if you will see here, that enzyme have some structure. So this is also known as the lock and key hypothesis about the enzyme and substrate. So let's say this is kind of lock and substrate is known as key, then it will fix. Key will be fixed into lock, and then this bond will be break down, and this substrate will be break down into product, that is another product, let's say A and B, and this enzyme will be detached from that product. Okay, and if you will see, this structure is very much different from this structure. Here the structure is this one, right? This one and this one. So we need different type of substrate here, and this different type of substrate will form a complex and will form another product. So enzymes are very specific with the substrate, and the substrate will have different shape. For that shape we do have different shape enzyme. Okay, and this is known as lock and key hypothesis or theory about the enzyme substrate. Okay, so this is how this answer is the right one. Enzyme bound. So this answer is the right one. Enzyme bind to specific substrate based on their active shape. So B is the right answer. Okay, what happened when an enzyme is denatured? If you know that denatured is, let me give you some explanation about this. We have egg, and egg have a liquid part. This is a yellow in color, yellow liquid. But if we boil it into like high temperature, or let's say if we are changing the condition of this liquid of that egg, then this egg will be changed into the white. So this will be your omelette. So we have the liquid part, but this will be converted into white part. So this is something known as the denature. So we do have the protein, and if we are changing the temperature, then this protein can be break down and from that complex to the simpler a component. So this is known as the denature. And if you people do know that enzyme is also made up of the proteins, just like your egg yogurt, sorry, egg yolk. So if this egg yolk will be boiled, then this will be changed into denatured form, or this is the white form. So likewise, if we are giving some temperature, or if we are changing the pH, then this protein based enzyme will also be denatured and will change into shape or maybe another thing. So based on that, we have question: what happens when an enzyme is denatured? It become more efficient, it loses its shape and function, it speed up the reaction in definitely, and it changes the substrate into a different enzyme. So based on explanation, yes, B is the right answer. So if you will see here, initially we had the enzyme and we had the substrate, and if we are changing the temperature and pH, then this enzyme may change the shape. Okay, and if we do have the optimum, then this will have the same shape as it had once. So this is how this is denatured. Denatured means we had the complex protein structure, and if we are breaking down this complex protein structure into the simpler structure, this will lead to the change in shape, change in color, and this is known as the denatured. So many of like people say, which one is better, egg yolk without boiling or after boiling? So this is like you people can search. White yolk is something after denature, right? Okay, the next question is the difference between virus and virions. So we have four options. Virion is an infectious virus outside a host cell, while a virus is only inside the host. Virus has a complete genome, while a virion does not. Virion is an incomplete form of a virus. Virus is made up of only protein, while a virion contains genetic material. So what do you think? What is the right answer? What's the difference between virus and virions? So the right answer is yes. So this is A. That if we have the virus can only reproduce when it have the host. So as I told you, virus only does have the genetic material, and that genetic material is covered with some capsid and some proteins. Okay, nothing that virus have. Virus is not the cellular organism, it is non-cellular organism, and it has to hijack the machinery of the host cell. So for example, human, if human are made up of cell, so virus have to hijack the machinery of our cell, that will help this virus to reproduce. Okay, so virus can't reproduce outside the host, but there is a one category, that is the virions. These are the infectious virus that can also survive outside the host cell, and that can also reproduce outside the host cell. So this is the basic difference between virus and virions. So that's why A answer is the right one. So this is the structure of virions. So we have genetic material, and this genetic material is covered by capsid. This is made up of protein and different component. Then these have the envelope, and we also have envelope protein and some enzyme. So this is just the thing that we have in the virus or virions. So it doesn't have a very organized cell. It is just like, if you are defining the virus, so we can define it as it is just the package of genetic material. It is not cellular. This is just this is just the genetic material that has been covered with some layer, capsid and some envelope. So this is how we can explain virus and how it is different from virions. That virions can reproduce outside the host cell, but virus can't reproduce outside the host cell. So this is how we can differentiate. Okay, why are virus considered obligate intracellular parasite? So we have four options. They can reproduce on their own. They can grow and metabolize outside the host. They require a host cell for replication. They can generate their own energy. So what do you think? What is the right answer? What do you mean by obligate? Obligate intracellular parasite. Based on that, you can give me the answer. Obligate, yes. So C is the right answer. So obligate means very necessary, mandatory. So these are mandatory, means they do not have another choice for the reproduction. For reproduction, they have to be intra, means within, cell, parasite. They have to be parasite inside the host cell. It is very necessary for them. It is mandatory for them. They can't reproduce outside the host cell. So that's why they are also considered as obligate intracellular parasite. So because they require host cell for the replication. So that's why D answer is the right one. Sorry, C answer is the right one. And let me show you, because we talked about reproduction, so let me tell you how they hijack the machinery of the host cell. So this was the let's say this was your virus. Okay, then, and this have just the genetic material here, and it doesn't have any machinery to reproduce or to have the duplication of its genetic material. It have nothing. It just have the genetic material. It has like no ribosome. It doesn't have any like Golgi apparatus. It doesn't have anything. So it has to hijack the machinery of your host cell. So it will come inside, and this is receptor. This receptor will bind to the virus, and this genetic material will come under this. Okay, so now the genetic material is inside the host cell. Then this genetic material use the machinery of the host cell and will replicate. So it will duplicate its DNA or its genetic material. Then it will translate. Translate means based on the code that that it has on the genetic material, that code will be read, and based on that specific protein will be synthesized. So this process from genetic material to the synthesis of protein is known as translation. Okay, so translation, the building or synthesis of protein will be done based on the information that we have from the genetic material. So after using the machinery of host cell, this virus genetic material will be duplicated. This is known as the replication. After that replication, it will synthesize the protein that is needed for the virus to grow, to reproduce. And this is how then this will use the Golgi apparatus. This is the Golgi apparatus of the host cell to assemble the things. And then it will have another, it will have another genetic material. And because of that protein, as I told you, that capsid and the cover of your virus is made up of the protein. If it is manufacturing the protein and another component within the host cell, it will create another virus. Okay, and this is how, this is first virus, and then it will create another virus. So this is how, using the machinery of the host cell, the virus will be duplicated into the another one. And this is how this virus will infect a thousand of your host cell, and will use the machinery of your host cell, and will infect the host cell. So this is how like we can tell the mechanism how the virus use the machinery of your host cell. And this is, and the reason is, as I told you, this virus have nothing but just the genetic material. So that's how it has to use the machinery of host cell. So that's why this is obligate, means necessary or mandatory intracellular parasite. Okay. This is again the same thing, but in a different way. So this is like, your virus can be double, virus may have double standard DNA. Your virus may have single standard DNA. Your virus may have single standard RNA, and your virus may also have single standard RNA, but positive. But at the end, what it will do, it will create mRNA. It will create mRNA. It will translate into proteins, and that proteins and then genetic material will be used to give another viral, another virus. So this is how it will reproduce. Okay, so this is the explanation for this. Okay, what is the function of protein capsid in the virions? As I told you, we have the genetic material, and this genetic material, let's say this is the genetic material, and this genetic material will be covered with capsid, and then they have the envelope, and in that envelope they have some proteins that will help them to bind with the host cell. So, and this is capsid, right? This is capsid. So the question is, what is the function of protein capsid in the virions? So we have four options. It allows the virions to generate energy. It protects the viral genetic material and help in host cell recognition. It allows the virus to move actively toward a host, and these it is responsible for viral metabolism. But yes, if we are talking about the covering, the protein capsid, then no doubt it will protect the genetic material and help in host cell recognition. So that's why, yes, your answer is right. That is B. Let me show you here. This is your capsid protein. So this is your genetic material, this is your capsid protein, and then you have another dimers. So this will help you to protect the genetic material in virus and virions. So this is the capsid protein. Okay, next question is, what is the key difference between a naked virus and enveloped virus? So we have four options. Enveloped virus have an additional lipid membrane derived from the host. Naked virus contain a protein coat, while enveloped virus do not. Naked virus are more infectious than enveloped virus, and D is enveloped virus lacked genetic material. Let me tell you, D is not the right answer because virus is nothing but the package of genetic material. So we have to have this genetic material. So this is not the right answer. Then you have the answer only from A, B, C. So what do you think? What is the right answer for this? So A is the right answer. If we have the enveloped virus that have additional lipid membrane and that has been derived from the host. So let me tell you with the figure. So you have naked virus and enveloped virus. So you have genetic material and you have the capsid. We talked about this. Capsid will protect the genetic material, but and this is the naked virus. But if you have enveloped virus, then you have some envelope, and this envelope has been generated from the host. So this is how it has additional layer of lipid. Okay, so this is how we can differentiate between naked virus and enveloped virus. Here also you can see some example of enveloped and non-enveloped virus. So if it is enveloped, then the example may be harp virus, it may be retrovirus. And if we are talking about non-enveloped, then this can be adenovirus and the parvovirus. A lot of example we have that can categorize viruses into enveloped virus and non-enveloped virus. Okay, next question is, how does a virion infect a new host cell? So we have four options: by dividing through binary fission. B is by using cellular machinery to produce its own energy. C is by binding to specific receptor on the host cell and injecting its genetic material. And D is by fusing with another virion to form a larger virus. What do you think? What is the right answer? How virion infect a new host cell? So if let's say we human is the host cell, and if virion needs to infect, do the infection on this virus, sorry, on the human cell, then how it will infect the human cell? So let's say this is the human cell and this is virion. So how this virion will infect the human cell? So what it will have? It have some protein, right? It have some protein, and this human cell also have some receptors. So what will be happen? This virions will be bind to the receptor, and then after getting the reception by the host cell, this will go inside and this will inject is genetic material. Let me show you how. So that's why B is the right, C is the right answer. Let me show you how. Okay, I do not have this, but I had one figure. Here it was, no? This was the host. This was the host membrane. This was the virus or virions. So it has the genetic material. It will inject this genetic material. So this is the genetic material of the virus or virions. So this will first will be received by the host cell, and then it will transfer or inject its genetic material into the host cell. So this is how it can infect. Let me go back to where we were. So that is why C is the right answer. Okay, now we are differentiating between autotrophs and heterotrophs. In autotrophs and heterotrophs, we have different categories: photoautotrophs and chemoautotroph. And likewise, in heterotroph, we also have chemoheterotrophs. So based on that, we have the question: which of the following best describe a chemoautotroph? Then we have four options: uses light as an energy source and produces its own food, These is uses chemical as an energy source and produce its own food. C is uses chemical for the energy but depends on another organism for food. And D is uses light for energy and depends on another organism for food. Okay, so you have to see, chemo means it needs some chemicals. Okay, auto means it will create or it will generate its food itself. Okay, so it had not to take the food from another organism or from another organic material, but it will create its own food. So that's why this is auto. And chemo means it needs some chemical to synthesize its own food. So that's why B answer is the right one. And let me show you the difference. Yes, Ramya, B is the right answer. So if we will see, we do have four categories. Photoautotroph. Photo means light, so energy source should be light. Auto means it will create or synthesize its food itself, but the carbon source will be carbon dioxide. So this is just like the plants. So plants uses the light and the carbon dioxide to synthesize its food, that is the cellulose, sorry, the glucose. So in the presence of sunlight, CO2 will be changed into glucose and oxygen. So these are known as the phototroph. If we have the chemical autotroph, means instead of light, it will use inorganic chemicals. Okay, and the carbon source will be CO2. And how this will synthesize its food, but in the presence of inorganic chemicals, not in the presence of light. So for example, if we have sulfur oxidizing bacteria, so sulfur oxidizing bacteria, what they will do, they have inorganic chemical, and they will have the carbon source, and this is how they will synthesize their food theirself. Okay, then we have photo heterotroph. Hetero means they can't produce their food itself, but they have to take it from outside. So for example, we can't synthesize our food inside our body. We have to take it from outside. We are eating plants, we are eating animals to have our carbon source. So our carbon source will be any organic chemical. So for example, grass if we are eating, or let's say if we are eating chicken, then this will be our carbon source. But we are not photoheterotroph, we are chemoheterotroph. But in photo heterotroph, the energy source is light, and we humans are chemoheterotroph because we are using organic chemical for the energy source as well as for the carbon source. So this is how we can differentiate between photoautotroph, chemoautotroph, photoheterotroph, and chemoheterotroph. Right. Next question, I think you can tell very exactly. So how do chemoheterotroph different from photo autotroph? So hetero to auto, and here chemo to photo. So A is chemoheterotroph, uses light for energy, while photoautotroph use chemicals. Then B is chemoheterotroph require organic compound for energy, while photoautotroph make their own food using sunlight. C is both rely on energy, light energy, but differ in their carbon sources. And D is chemoheterotroph make their own food, while photoautotroph must consume another organism. So if we will see here, auto means they will synthesize their food themselves. So must consume another organism is not the right statement. So this is not right. Okay, and as I told you, one is chemo and one is photo. Photo means light. So this photo autotroph needs light as an energy source, but here chemo heterotroph need chemical for the energy source. So both are not dependent on the light. Chemo is dependent on chemical, photo is dependent on light. So this is again not the right answer. Then as I told you, for chemo we need chemicals, We do not need light, so this is not the right answer. So the remaining one is B. So B is the right answer, and we already have talked here, no? Energy source, when it is photo means light. When it is chemo means inorganic chemical. And when we are talking about heterotroph, hetero means they do not just need CO2, but they need organic chemical for having the carbon source. Okay. Which of the following is an example of a chemoheterotrophic organism? So we have green plants, cyanobacteria, fungi, and sulfur oxidizing bacteria. So what do you think? What is the right answer for this? A, B, C, D. If we are talking about the green plants are autotroph. Auto means self. So they produce their food themselves. So these are not the hetero. So this is not the right answer. Cyno is also have the chloroplast, or you can say uh, some of the component in the cell that can produce, that can do the photosynthesis. Photosynthesis means in the presence of light, they can they can synthesize their own food. So these are also autotroph, but not the heterotroph. Fungi, yes, fungi need inorganic compounds for energy source as well as for the carbon source. So C is the right answer. And if we are talking the sulfur oxidizing bacteria, then these are chemoautotroph. These are not hetero, but they can synthesize their own food. But in spite of photo or light, they should have the chemical, inorganic chemical. So that's why C is the right answer. Okay, why can't chemoheterotroph survive without other organisms? So we have four options. They depend on another organism for both food and carbon. They generate energy from inorganic source. They can perform photosynthesis. They do not need organic molecule to grow. What do you think? What is the right answer? So chemoheterotroph. Hetero means they do not synthesize their own food. For example, we human, okay, our body cannot synthesize their own food, but we have to have it from outside. Okay, so and without another organism, so we need food, right? And from where this food will come? This food either will come from plants, or let's say organism and animals, or from marine organism. So we need another organism on which we are dependent. So that's why A is the right answer. We can't use inorganic source, we have to use organic source. For example, if we are having chicken, or let's say if we are having the salad, that salad or chicken have organic source of energy or organic carbon source. So this is not the inorganic, so this is not the right answer. We can't perform photosynthesis, and we need organic molecules to grow. So do not means this is not the right statement. So the only option is they depend on another organism for both energy and carbon. So that's why A is the right answer. I already have given you the explanation for this, so I don't think that I should go deeper into that. Okay, which of the following organism is not classified as a chemoheterotroph? Human, fungi, algae, most bacteria. Hetero means they can't synthesize their own food. So, and it is not classified as chemoheterotroph. So we are talking about the autotroph. So which of these are autotroph in nature? Which of the organism that can synthesize their own food? Bacteria can't synthesize their own food. They must have some decay organic material or some inorganic material from where it can do something. But yes, algae can do photosynthesis. Photosynthesis means in the presence of light, in the presence of carbon dioxide, they can create their own food. So algae is the right answer. Algae is not the chemoheterotroph, but it is the photoautotroph. Algae is the photoautotroph. Photo light, in the presence of light. Auto means self food. So they can create their own food in the presence of sunlight. So that's why C is the right answer. Yeah, so these are some of the algae. I think like if you are biology student, then you must have seen into your laboratory in the microscope. So these are different type of algae that we have. Okay, so according to the binomial nomenclature system, how should a bacterial scientific name be correctly written? So we have four options: the genus and the species names both in uppercase, the genus capitalized and the species in lowercase, both italicized. Then C is the genus is lowercase and the species in uppercase, and D is both name written in lowercase without italics. Yes, so yes, B is the right answer. So binomial means two. Okay, so if we are the scientific name always have two component. The first component is genus and second component is species. First component has to be capital in nature, and the species should have the lowercase. So let me show you some of the example. So, for example, if we have the tiger and the tiger name is Panthera tigris. This Panthera is genus name and this tigris is the species name. And if you will see here, P is capital. P is capital and T is lowercase. Okay, so and this is how there is a rule of the binomial classification that the genus must have the capital or uppercase of its first letter and the species should have the lowercase. If it is an elephant, then also Loxodonta and Africana. Here Africana has A, that is lowercase, and Loxodonta has L, that is capital. So this is the rule of the binomial nomenclature, and that has been given by Carolus Linnaeus. Okay, that was scientist that started this binomial nomenclature. Okay, what does the species name in bacterial nomenclature indicate? The physical shape of the bacterium, the ecological niche or host where the bacteria was found, the antibiotic resistance of the bacterium, the classification kingdom of the bacterium. So we have like two component in the naming, scientific naming, and the first is sorry, I use that thumb and that emojis generated itself. So uh, so we have two, the species and genus and species, and you have to tell me what does that genus means or species means. No, D is not the right answer, but B is. Let me show you how. Just see here. If it is Loxodonta africana, so Loxodonta means uh, elephant. So we have different type of elephant, right? We have different species, but that different species is very different in different biogeographical zone. So this elephant is present or is can be found in the Africa. So this is Africana. So what it is showing, we have the genus name, but if we do have the species name, that species name is telling where it can live, or from where it is found. So this is talking about the host, or it is talking about the location where it is present abundantly. So the species is for the specification about the host or the place where it originated or from where it is found. So for example, if I give you the name of mango, this one species is Mangifera indica. So indica means this particular mango species is found in India. Okay, and if it is Africana, means it is found from Africa. So I think that's how it is clear. Then the species name indicate the ecological niche. Niche means this is not very much similar to the habitat, but right now you can just think that this is habitat from where the bacterium or any organism was found. So that's why B is the right answer, not D. This classification kingdom of the bacteria, no, this is not the right answer. B is the right answer. I think this is clear now. Why? Just take the example of Menzifera indica. So we have mango species. If it is in India, then that species will be known as Menzifera indica, and indica means the habitat or that location from where that species was found. Okay, so here if we will see, I'm not able to see my slide. Are you people able to? Okay. Why is Latin used for the bacterial nomenclature? So let's say it is Menzifera indica, or it is like for rice, the name is Oryziva sativa. Then this is Latin. The language is used in this nomenclature is Latin. So why we people use this Latin language only? So I have four options, out of which you have to tell me. Latin is the official language of microbiology. Latin is universally understood and do not change over time. Latin is easier to pronounce than another language, and Latin name indicates the geographical origin of bacteria. So, what do you think? What is the right answer? Why we do Latin for the nomenclature? So the answer is yes, this is B, that this is universally understood and do not change over the time. And because it do not change over time, that's why it is also known as the dead language. Let me show you. So this binomial system uses the Latin language because it is the dead language. It will no longer change. Every country uses this language, no matter what language they speak, because this is that language. This will not change over time. And scientists use the genus and species in Latin language only. So if it is Africana, if it is Indica, then these are Latin words. Okay, the next is, which of the following structure is present in prokaryote cell but absent in eukaryote? We already have talked much about the difference between prokaryotic and eukaryotic, but this is something about the organelles. So in prokaryotic we do have something inside the cell, in eukaryotic we have something inside the cell, and you have to tell me which structure is present in prokaryotes. Just see, in prokaryotes but absent in eukaryote. So the options are mitochondria, nucleolus, ribosome, and cell wall. If Smita, you are changing your answer. Initially you told me A and then B. Are you like confirm your answer B, or you want to change it to A again? Anyway, so D is the right. If Smita, D is the right answer. Uh, okay, so because here it was something trickier, because I talked about the present in prokaryote but absence in eukaryote animal cell. In the plant cell, we have the cell wall, but if we have the animal cell and that is eukaryotic in nature, then that animal cell do not have the cell wall. They just have the plasma membrane. Okay, and the question was, I know that question was little trickier. If it was absent in eukaryotic, and that eukaryotic maybe plant cell and animal cell, then you can give the answer different way. But because it was eukaryotic animal cell, then in an animal cell we do not have the cell wall. But in prokaryote, we talked about that. In prokaryote, in bacteria, we have peptidoglycan cell wall, right? In eukaryote as well we have the cell wall, but in fungi, as I told you, in fungi we have the chitin based cell wall. In plants we have cellulose based cell wall. But if we are talking about the eukaryotic animal cell, then cell wall is not present in the animal cell. So that's why D answer is the right one. Okay, is it clear, Smita? Okay. So here, if I show you the prokaryote cell, we have the cell wall, outermost layer of the bacteria. If it is the plant, then also it have the cell wall protecting the plant cell. But if we do have the animal cell, then it just have the plasma cell membrane or plasma membrane. It doesn't have the cell wall. So based on that answer, that eukaryotic animal cell doesn't have the cell wall will be the right answer. Okay. Which of the following best describe the genetic material of a prokaryotic cell? Linear chromosome enclosed in a nucleus, circular DNA floating freely in a cytoplasm, DNA enclosed in a membrane bound nucleolus, and DNA stored in mitochondria. What do you think? What is the right answer? We talked about this. Pro means false or very primitive karyotic nucleus. So here in prokaryotics, we do not have the true nucleus. So genetic material is just scattered into cytoplasm. But in eukaryote, U means true, karyotic means nucleus. So we have true nucleus, and in that true nucleus, we have the genetic material. So you gave me answer B, then answer B is right. In prokaryotics, the DNA is mostly circular in nature, but in eukaryotics, genetic material is linear. Okay, and these, as I told you, prokaryotic means primitive nucleus. So that's why the genetic material is floating freely into the cytoplasm. So that's why B is the right answer. This linear chromosome enclosed in a nucleus, it is the case of eukaryotics. So this is not the case of prokaryote. If we are going to the C, DNA enclosed in the membrane bound nucleolus, this is again not the case of prokaryotes. And DNA stored in mitochondria, this is the perfect case of eukaryotics, where in mitochondria we also have the DNA. So that's why B answer is the right one. So here, if you will see, this is the bacterial cell. And in that bacterial cell, we have the nucleoid, and this is also known as the chromosomal DNA. This is circular double stranded piece of DNA, not surrounding by the nuclear membrane. And they also have plasmid, and these plasmid are independent DNA molecules. So they have two type of genetic material. One is the nucleoid that have chromosomal DNA, and the another is plasmid that have independent DNA molecules. So this is how we can talk about this. Okay, which organelle is present in both prokaryote and eukaryotic cell? Mitochondria, ribosome, Golgi apparatus, and endoplasmic reticulum. Epsmita is very fast giving me the answer B, then obviously this should be B if you are like confident. Then yes, let me show you here. We have prokaryote cell and eukaryote cell, and if you will see here, rhizosome is the organelle that is common in both, and the work of the rhizosome is to help in the synthesis of the protein. So prokaryote and eukaryote also need some organelles that can help in the synthesis of protein, so both have the ribosomes. Okay, both have the plasma membrane and the cytoplasm. Cytoplasm is the place where all the organelles, for example, ER, that is endoplasmic reticulum, Golgi apparatus, nucleus, everything is present in the cytoplasm. So this is a liquid sac, you can say, and then in which ribosome is the common thing that we have in both prokaryotes and in eukaryotes. Okay. Then why do prokaryote cell not undergo mitosis? They lack DNA, they divide by binary fission instead. They do not have the plasma membrane. Their DNA is stored in the mitochondria. Let me tell you mitosis. So mitosis is kind of division. So if we have one cell, that one cell will be divided into two cells, and this is with the help of mitosis. This process is mitosis, in which so one cell will have some chromosome or the genetic material. That genetic material will be duplicated, and that duplicated material will be go into another cell. So in humans, if we have one cell and this cell is divided into two, then this process is known as the mitosis. And this mitosis is not comes under for prokaryotic cell, but only for the eukaryotic cell. Why? This mitosis is a little complex as comparison to the binary fission. So most of the prokaryote cell undergoes into binary fission. So binary fission means they will divide as a, let me show you. So B is the right answer. So this is how the cell division has been happened. So they have genetic material, they have one cell, then this genetic material will be replicated, then we will have two genetic material, and then they will this cell will undergo into the fission means breaking down. So this cell will be break down into two cells. So this is the binary fission of the prokaryotic cell division. And for eukaryotic, binary fission is not happening, but we undergo through mitosis. So through mitosis, the cell division is happen in the eukaryotics. Okay, so that's why this binary fission answer is the right one. Okay, which of the following is the key characteristic of the eukaryotic genome but not the prokaryotic genome? So we have four options: presence of nuclear chromosome, presence of multiple linear chromosome, DNA located in a nucleoid, genes organized in operons. Key characteristics of the eukaryotic genome but not the prokaryotic genome. What do you think? What is the right answer for this? Okay, so if we are talking, no, in prokaryotes we have nucleoid, but in eukaryote we have nucleus, and in that nucleus we have nucleolus. And we are talking about the key characteristics of the eukaryotic genome. And if, let me tell you, if this presence of circular chromosomes are not present in eukaryotes, but these are present in prokaryotes. So A is not the right answer. Presence of multiple linear chromosome, yes, this may be answer, because eukaryote have the if you people know that we do have 46 chromosome, and like it is divided to 23, so 22 plus 1, so we have 22 chromosome plus 1 autosomal 22 chromosome and 1 6 chromosome, and if they will duplicate, then this will be into 46. Okay, so and we do have a linear chromosome, then this may be answered. Then DNA located in a nucleoid. DNA is located in nucleoid in prokaryotic genomes, not in eukaryotic. And genes organized in operons. So I will tell about this. This operon characteristic is of the prokaryotes, not the eukaryotes. And that's why the multiple linear chromosome answer is the right one. That is the key characteristics of the eukaryotes, you know. So let me show you here. So if this is the prokaryote, here you can see the bacterial DNA and plasmid. These are circular in nature. And if we are talking about the chromosome that we have in the multicellular or eukaryotes, then we have the nucleus. In that nucleus, we have chromosome, and that chromosome have the DNA part, and this DNA is linear. And we have multiple chromosomes. So we do have linear multiple chromosome. Okay, so this is the characteristic of the eukaryotes. And this so we have in the chromosome, we just do not have DNA, but we also have some protein. To that protein, DNA is binding, and that protein is known as histone protein, and this histone protein is not present in the prokaryotic chromosome. Okay, and here in prokaryotic, they just have one chromosome, but in eukaryotes, we may have two or more different chromosomes. So for example, in humans, we have 22 plus 1, right? And it is in pair, then we can say like this is 46. So just keep in mind, in bacteria, we have circular DNA. In eukaryote, we have linear. Uh, in bacteria, we do not have the proteins that is binding to the DNA, but in eukaryotics, histone protein is binding the DNA. And in eukaryotic, we have multiple chromosome, but in prokaryote, we do not have multiple, we just have one chromosome. Okay, so this is how we can give the answer for this. Okay, how does gene regulation in prokaryotic primarily differ from that in eukaryotes? So we have four options. Prokaryotic genes are regulated individually. Eukaryotic cells are organized in operons. Prokaryotic genes are often grouped into operons, and prokaryotic cell use histone for DNA packaging. So let me first tell you, we have cell. In that cell, we have nucleus. Okay, I'm talking about eukaryotic. Okay, so we have cell. In that cell, we have the nucleus. In that nucleus, we have chromosome. And in that chromosome, we have DNA with the proteins. Okay, and that DNA is very long in nature. And that DNA, some segment of DNA is known as gene. Okay, and that gene can that gene will be that gene first will translate into mRNA, and then after mRNA, that can synthesize some specific type of proteins that we need in our body. Okay, so cell has the nucleus has the chromosome have the DNA, and the segment of DNA is known as the gene. And that segment of DNA have some different parts. Okay, and that part I will tell you in the next slides. So here some part may regulate the genes. Okay, so the question here is how the gene regulation is prokaryotically primarily differ from eukaryotic is that prokaryotic gene are often grouped into operon. Let me show you how. So as I told you, one segment of the DNA is known as gene. Okay, so we have, so let's say this is the DNA. the linear layer, and we have gene 1, gene 2, gene 3, gene 4. But before that, uh, we have some segment of the DNA that have A, that is the activator protein binding site. We also have P, promoter, and O, operator. So what will happen when some protein will bind to this A? Then this gene 1, gene 2, gene 3, gene 4 will start to replicate, will start to duplicate its copy. But when the protein will bind to, let's say, O, then the replication will be stopped. So then you can see, and this promoter, operator, and genetic structure are known as the operons. So the group of some genes are known as operons on bacterial cell. So in bacterial genetic material, this APO will regulate, these are the this will regulate the genetic activity of the bacteria. So that's why these are grouped in the operands. But this is not the case in eukaryote. In eukaryote we have different mechanism. But just in keep mind that in prokaryotics we have the operands. Operands means some of the genes are grouped together, and this will regulate the genetic activities. So for example, activator will activate the genetic activity, O will so when some protein will bind to this O, then this genetic replication will be stopped. So this is how this is operands. So this is the key characteristic feature of prokaryotes, but not the eukaryotes. So that's why this one is the right answer. Prokaryotic genes are often grouped into operons. So I think this is clear. I consumed a lot of time in this so that you people can understand this concept. Uh, okay. And as I told you, we have some promoter, operators, and some genes. And these genes, first we have the DNA. This DNA information will be translated, will be transcribed into mRNA, and this based on mRNA, some protein will be formed. Okay, and that we need in the cell, and this process is known as translation. So we have DNA to RNA is known as transcription. From RNA to protein, this process is known as the translation. Okay, which is the major structural difference between eukaryotic and prokaryotic chromosomes? We are talking about chromosomes here. So eukaryotic chromosomes are circular. I don't think that this is the case, because eukaryotic chromosomes are linear in nature. Eukaryotic chromosomes are associated with histone protein. I told you that in the eukaryotic we have chromosome, and this chromosome also have protein and the DNA, and that protein is the histone protein. This may be the case. Then prokaryotic chromosome have multiple origin of replication, while eukaryotic chromosome have only one. This may or may not be the case. I will explain later. Then prokaryotic chromosome contain repetitive sequence like telomere. This is the case in eukaryotics. Eukaryotes have the repetitive genes, or you can repetitive sequence. And that's why we have a very large DNA, and that have the repetition of some of the codes, and that will help us from the mutation. So this is not the case of prokaryote, but this is the case of eukaryote. So now you can tell me what may be the right answer. So B is the right answer. Yes. Again, the same figure as I told you. We have circular DNA and very freely stay in the prokaryote, but in eukaryote, in chromosome, we have the DNA, but it is bind with the protein. That protein is known as the histone. So that's why B is the right answer. Okay. Which of the following statement about plasmid is true? As I told you, here we have the bacterial cell. In bacterial cell, we have the bacterial DNA, and we also have the plasmid. So plasmid is also some kind of genetic material that we have in the bacterial cell. So that next question is based on plasmid only. So plasmid are linear DNA found in eukaryotic cell. Plasmid are small circular DNA molecule that replicate independently. Plasmid contain essential genes for bacterial survival. Plasmid are found in eukaryotic nucleus. So what do you think? What is the right answer? First option is not right, because as I showed you, here the plasmid is circular in nature, and this is present in prokaryotes, not in eukaryote. So that's why A is not the right answer. D is also not the right answer, because it is present in prokaryotes. Then plasmid contain essential gene for bacterial survival. This is not that much case, because all the genes that we have in proper that chromosomal DNA, it also have. But the B answer is the right one, that is have the small circular DNA that replicate independently. Okay. So here also you can see, this is the bacterial and this is the plasmid, and this plasmid can replicate independently because it has a lot of things. For example, it has promoter, it has markers, it have some genes, and origin of replication, so it can replicate itself independently. Okay, which of the following is found in eukaryotic genome but not in prokaryotic genome? So as I talked about, the eukaryotic or human genome is very long. And why this is very long? Because it have some genes that are not necessary. It have a redundancy in the sequencing of the human genome or eukaryotic genomes. So that part of the genes that are repetitive in nature, that are not necessary for the synthesis of the proteins, are present in eukaryotic but not the prokaryotic, and these parts are known as introns. Okay, so let me show you how. So B is the right answer, and let me show you. So we have the gene, and in that gene we have some sequence that are known as exon and some sequence that are known as the introns. And whenever from DNA we are transcripting into mRNA, then we have to remove these introns because these are unnecessary. Okay, not unnecessary, these are necessary but not for the mRNA. These are necessary to stabilize the DNA. These help to avoid the mutations. So these are necessary, but in mRNA we do not need these introns, but we need just the extron. And this is the property of eukaryotic chromosome only, not the prokaryotic. So that's why this one gives you the right answer. Here you can also see, like we have some coding region in the DNA, and in that coding region also we have exon, intron, and we have to remove this intron to have just the extron for the transcription. Okay, time is up, but let me just cover 3 or 4 questions more. Which enzyme is responsible for synthesizing complementary DNA from an RNA template? So, for example, if some bacteria have the genetic material that is RNA, okay, that is not DNA, and if you are having complementary DNA from RNA, then which of the enzyme will help it to do this? So some gave me C answer and some gave me D answer, but C is the right answer. Why? Let me tell you why. Because most of the time what we do, we have the DNA, and from DNA we have the process. That process will build RNA. So the thing is DNA to RNA to protein. But if we are having RNA to DNA, we are doing the reversing. So based on that, we need the enzyme that is the reverse transcriptase. Okay, so most of the time it is DNA, RNA, protein. The sequence is like this. But if we have RNA and then we are going to the DNA, then we need enzyme that is the reverse transcriptase. And this is how we have virus RNA. Then based on this enzyme, we can start to have the complementary DNA. and then we have the double stranded DNA. So we need enzyme that is reverse transcriptase. Okay, this is very logical question. Why is complementary DNA used instead of genomic DNA for cloning eukaryotic genes in prokaryotics? So because we do not have time much, so let me tell you that as I tell you, our DNA, that DNA we have in the human, that have intron, exon, intron, exon, intron, exon, and introns are the unnecessary that we have to remove. We just need to have the extron in our mRNA. Okay, and if we are going from mRNA to DNA, then this extron are removed. Okay, so that's sorry, introns are removed. So that's why if we are having from RNA to complementary DNA, then we do not have the introns in our DNA. So that's why this give us the beneficial feature. So cDNA is free of introns, making it easier, because as I told you, if you remember this, let me show you. So this one, so we have DNA, and if we are going mRNA, then these introns will be removed. And if we are going from mRNA to DNA, then this is free of introns. So that's why C is the right answer. This is again, I don't think that in very short period you will understand this, but just keep in mind we have RNA, and from RNA we have like initially if we have like DNA to RNA, then we need polymerase, and initially we have exon, intron, exon, intron, but it will be spliced, it will be removed, and then we will have only exon, exon. So, and if we are doing the reversing of this, then our DNA will not have the introns. I think I should skip right now some questions. This is not the. I'm skipping. Okay, this is important and very simple question. Which of the following best describe a genome? What do you mean by genome? So genome is the entire set of DNA that also include coding and non-coding. We talked about non-coding because introns are not coding region, that are non-coding region. So if we are describing the genome, so then genome is entire DNA that have coding and non-coding region. Let me show you. So in that chromosome we have genetic material, that is DNA, and in that DNA we have some region that can be coded and in another we can't. So this is how this whole region, this whole DNA will give you the genome, right? Okay, which component of the genome is responsible for coding functional protein? As I told you, introns will be removed, but exon will give you the functional proteins. We talked about it a lot, right? And here, this introns will be removed, spliced, and then after that in mRNA, we will just have the exons. So this will give you the code for the proteins. Then we have mitochondrial gene as well, and in the nuclear gene in our eukaryotic cell. So for example, in humans we also have mitochondrial genome. In the mitochondria we also have the genetic material, and this is different from nuclear genome, is that this mitochondrial genome is circular in nature. So it is said that once like millions billions year ago, the bacterial cell entered into our cell, so that mitochondria is something like bacterial cell, and that have this own circular chromosome or circular genomic material. So that's why this mitochondria, this is theory. So this mitochondrial genome is circular in nature and very smaller, so that's how it is different from the nuclear genomes. And here you can see the description, because I will upload these slides on the link that has been given, so you people can either watch this video again, or you can also like access these slides from there. So because right now I don't have time, I am just covering a few questions here. Which of the following statement about non-coding DNA is true? And because some genes will be used for the coding of the proteins, but some are non-coding, and this non-coding will regulate the gene expression, right? So that's why B is the right answer. Then we have two type of genomes now: nuclear genome and the organelles. So for example, mitochondrial genomes, we also have chloroplast genome in the plant cell. Okay, so we have to tell, like in the plant genome we have chloroplast genome and also as the nuclear genome. So plant cell have both. You can watch this. Then you have to tell me the organelle or that component of the cell of the plant that can convert sunlight into energy. So this is chloroplast. Okay, you can read about this as well. Then we have the vacuole in plant cell, and that vacuole regulate the turgor pressure. So that's why B is the right answer. Then one thing that we have in the animal cell but not in the plant cell, that is the centrioles. You can read about this. This help in the cell division. And if we do have the two cell, and how this two cell will communicate with each other, this is through the plasmodesma or plasmodesmata. So this is how C is the right answer. Then the virus can infect the bacteria, and that viruses that can infect the bacteria are known as the bacteriophage. And that's why, this is the reason why Ganga is so sacred, because whatever bacteria that Ganga have, the Ganga river also have 1,000 of bacteriophage. That is the viruses, that viruses can eat the bacteria. So that's why Ganga river is so sacred in nature. It can't give you the infection, because bacteria are removed by the viruses that are known as the bacteriophages. The bacteriophages that can eat the bacteria are known as the bacteriophage viruses. So that's how river is so sacred. Then we have some bacteria that can thrive in a very extreme condition. So for example, that can thrive in a very high acidic condition. So that bacteria, one of the bacteria is ferroplasma. And some of the bacteria may be very useful, and that may be found in human gut, and that may help in digestion. The one of the bacteria is lactobacillus acidophilus. If you people know that whenever you have the dysentery like condition, then doctor suggests you to have this lactobacillus tablet. So that lactobacillus tablet is nothing but thousands of that spores of the bacteria that are good for our digestion. So this is how B is the right answer, and that's how I completed my slide. I know I'm late, but this is how like it was the introductory session for the microbiology. If you have any query, you can tell me. Otherwise, you people can leave because this is the dinner time. I also have to go to mess. So thank you so much. If you have any query, you can write me in the chat box. So thank you so much, everyone. If you have any query, you can write. Thank you, everyone. Naveen, Radhika, Alekhya. Yeah, thank you, Dilip Kumar. Thank you, Ganendra. If you have any query, you can ask me, and you can also write anytime on my email ID, that is jyoti21@iiserb.ac.in. So you can anytime write in my email ID so that I can give you the answer. I will again meet you from 6 to 8 next Monday. Okay, so thank you so much.